Oil and water don't mix, but more importantly, oil and charged atoms don't mix. When you shake a vial containing water, oil, and a red charged salt like cobalt chloride, the red ions remain exclusively trapped in the water. They physically cannot enter the oily layer floating on top. Every cell in your body is encased in a 40-angstrom thick lipid membrane that acts exactly like that oil. It serves as a biological wall, keeping the chemistry of life inside while blocking water and electrical ions from passing through the oily interior. In 1988, Peter Agre discovered the first exception to this wall, the aquaporin. It is a specialized protein channel that allows 1 billion water molecules through every second. As they pass, the channel rotates each molecule midstream, a maneuver that prevents protons from leaking through and disrupting the cell's internal chemistry. This discovery provided the first evidence that cells survive the barrier problem by using engineered molecular machines to move essential ingredients across the membrane. Electrical ions are even more difficult to transport. To power the NOVA system, a potassium channel must move 100 million ions per second. At that speed, it must also be precise enough to completely reject sodium ions, which are nearly identical but slightly smaller. This creates a paradox. High selectivity usually requires a filter that binds an atom tightly to verify its identity, which would logically make transport slow. Speed, on the other hand, requires a low-resistance path where atoms barely touch the filter at all. Resolving this contradiction required a 3D map of the channel at the atomic level. In the 1990s, obtaining a clear structure of a membrane protein was considered one of the most difficult challenges in structural biology. This mechanism is the physical requirement for the high-speed electrical signaling found in the human brain. Without a way to be both perfectly selective and lightning fast, the rapid firing of neurons would be physically impossible.